Our next speaker is Alexandre Marand. He is a professor, assistant professor at University of Michigan. Dr. Marand did his PhD at the University of Wisconsin before doing his postdoc at University of Georgia. And he was a NSF postdoc fellow and also received a K-99 award. And today, Dr. Marand is going to be talking about the effects of regulatory diversity on cell identity.
Awesome. Yeah, thank you. Thank you so much, Ben, for the intro and to the panel for inviting me. I'm super thrilled to be here. I don't know if I'll be able to top Elliot's presentation. I don't have any major paradigm shifting examples like that, but hopefully I can convince you that understanding cellular diversity at the level of transcription is important. So the major question that my lab is interested in is really just understanding mechanisms of transcriptional regulation. And we're not developmental biologists, but we like to use the developments, I guess, engine to generate cellular diversity to understand why and how differences in transcriptional regulation can drive new types of cells. So, anyways, this is the kind of the broad question of the lab is understanding how sequences, non-coding sequences, can drive gene expression and how those sequences also encode the different patterns of when a gene is transcribed, when a gene is turned off, as well as the timing and the spatial coordination of gene expression. So some of the things that we think about are the binding sites for transcription factors, also known as cis-regulatory elements, and how multiple transcription factors work together through long-range interactions or through proximal interactions to turn on or turn off gene expression. So this is sort of the main question of the lab. So when we think about regulatory diversity, this is sort of the crux of what we're interested in is really through two different flavors. The first is within an organism. And so we think about regulatory diversity in this context as being sort of the regulation of cis-regulatory elements or transcription factor binding sites in general, being regulated by nucleosome occupancy. So nucleosomes, when they bind to DNA, they include most transcription factors access to that underlying DNA sequence. And so those regulatory elements bound by nucleosomes tend to be inactive. And so this is sort of within an organism. So among different types of cells or different cell states, you can get differences in nucleosome occupancy and therefore differences in cis-regulatory activity. The second sort of definition that we think about a lot is really functional diversity. So how do sequence changes within non-coding DNA affect cis-regulatory activity, and how does that impact gene expression as well as the development of the organism and phenotypes that we can see and measure? So I'm mostly going to focus today, because I don't have a whole bunch of time on this second aspect of regulatory diversity. And so being as, we're really interested in using maize for several reasons, but the reason we use maize, one of the main reasons we use maize is because it has an extraordinary amount of genetic diversity among different accessions sort of geographically distributed across the world. So one of the things I did during my postdoc was to collect these individuals that are known as the 282 maize diversity panel. Only about 172 liked the conditions that we had them in the basement of the building in Bob's lab. So we're now calling this 172 diversity panel. But what I did was to collect, these materials and subject them to single nuclei genomics. So that's single cell ATAC-seq. So we're measuring accessibility as a proxy for regulatory activity, as well as single nuclei gene expression or single nuclei RNA sequencing. And so ultimately what we get if we combine, if we using this experimental setup is we get many different cell states being profiled. I should mention that we sampled around the shoot apical meristem to include both the above ground and below ground tissue, just about 1 inch. So we have a diverse types of cells present in the sample. And so we were essentially creating this large database of many different cell identities across many different genotypes. And so the idea is, what is the effect of this extant genetic variation on regulatory activity within different cellular contexts? Okay, so you might imagine that, what you can do is, because we have these different genotypes, these different inbred individuals, we can compare and contrast the accessibility profiles among them. Okay, so here's an example of an accessible region that shows variation amongst the individuals in this panel. And as you might imagine, just like any other quantitative trait, we can map it, right? So here's an example of a Manhattan plot of this particular phenotype shown on the left and the variants that are associated with it. All right, so these are very strong. We can see a very strong peak of enrichment, of significance, of variants that are associated with this variation, okay, in cis. Okay, so here's just one of the examples of the lead variant. We can see that if you're homozygous for cytosine, you have very high levels of accessibility, whereas if you're homozygous for thymine, you tend to have much lower levels of accessibility. And so to sort of, convince ourselves that, what we're seeing is biologically relevant, you can look at where the chromatin QTL are located, and they're very strongly enriched within the accessible regions that they're associated with, right? So if we have variants within an accessible region, they tend to be associated with variation of that regulatory region. And because there was already existing data from this population in terms of measuring transcriptomes, we can also compare the known expression QTL with our chromatin QTL. And we can see that chromatin QTL are highly enriched also for affecting transcription, whereas non-chromatin QTL are depleted of effects on expression. So you can also look at how those effects are distributed, and we see about an 85% concordance between the accessibility variation. So when an alternate allele decreases, accessibility is associated with a loss of accessible chromatin. You also tend to see a loss of gene expression of that cognate allele. So we feel pretty good that we're capturing realistic effects where individual polymorphisms are affecting chromatin structure as well as gene expression. And as you might imagine, because we did single cell, we can also look at the cellular diversity within this panel. And so here's just an example of the different cell types or cell states. Later, I'm going to argue why we should kind of ignore, or try not to define cell states, but I'll focus really just on how we define them in this particular example by doing the same things that we have been doing over the past 5 or 6 years, which is to look at marker genes. And some of the other things that we also will use is these predictive models or regularized linear regression models where we can train a model to learn what is the gene expression of a given cell type. Of course, the logic is somewhat circular there, but I think those models are now becoming more refined and helpful to help us really identify what are the core marker genes that are really useful for defining a cell type, and maybe why defining a cell type is still useful. Okay, and so because we have single cell data and we resolve the different cellular contexts, one thing you can ask was, okay, well, what is the contribution of cell identity towards understanding or recognizing a particular genetic effect? So here's the example I showed just a couple of slides ago with the peak associated, or chromatin QTL associated with variation in accessibility for a particular region. All right, and so you see a strong peak in the bulk tissue, but we can split that by different cell contexts, right? Because we have accessibility information for each of these different cell types across the entire panel. So what you can see is that, yeah, we can find a strong peak within the bulk tissue, right? If we average all the cells together, you can see a signal, but an even stronger signal if we isolate just the mesophyll cells, or cells that have marker genes that are associated with mesophyll identity. Okay, but not, and that signal disappears in other cell states or other cell types. Okay, so that suggests to us that different cell contexts are going to interpret these genetic variants differently, right? So cell context plays a big role in how genetic variation is manifested within the cell. And also want to point out that, when we run this pipeline to identify chromatin QTL, we find many, 44%, that can't be identified in bulk tissue due to sensitivity. And so by parsing out the different cell types, we're able to capture more, you know, regions of the genome that show variation within a cell type among individuals that may not be present in other contexts. All right, so we're able to capture more genetic effects because we're able to parse out cells by their identity. Okay, so sort of collectively, if we take all this data together and ask, okay, well, how many of these chromatin QTL are shared among different cell types? And the answer is that almost none of them are actually shared across all the cells within a tissue. And so about 53% or more than half, the majority of chromatin QTL are found in 3 or fewer cell types or cell states in this example. All right. Even in those found in just a single cell type is about 16%, or it's a fairly large percent of all the QTL. And interestingly, those cell type specific QTL, right? So these are variants that only affect accessibility within a single cellular context, tend to also affect larger traits. So here's an example of a GWAS scan for plant height. Okay, so here's the typical minus log 10 p-value Manhattan plot for a given locus. And we can see that this signature co-localizes almost perfectly with the expression QTL as well as the chromatin QTL that was unique to the leaf primordia. And actually, I think this is a pretty interesting example for those of you or those of us who are analyzing ATAC-seq data. You know, we often will just compare the height of these peaks, but what's also interesting is that you can identify differences in the shape of peaks, which may also have, you know, some sort of biological importance. So here's an example of one where, you know, you don't get too much more accessibility if you're a genotype that's homozygous for guanine. Okay, but the region of accessibility expands, potentially opening up more regulatory sequences that are impacting the expression of this gene, which manifests into a change in plant height. Okay, so the takeaway here is that cell-specific QTL often co-localize with expression QTL as well as variants that are associated with agronomic traits of interest. All right, and then to sort of tie this up, the next thing we asked was, so we get these genetic variants that are affecting accessibility, but the question is how, right? So are they impacting transcription factor binding sites? Are they making it so nucleosomes are no longer able to load? So the first thing that we checked was, are there any particular transcription factor families where we find these chromatin QTL enriched? And so what we did was to basically, here I'm showing you in red the TCP family of transcription factors, of which there's about 24 members in maize. And across all the different cell states, we kept finding that chromatin QTL that overlapped or within those TCP binding sites tended to be overrepresented, regardless of the cell context that we looked at. Something else that we also found that was interesting, and I'm just putting leafy on here as sort of a reference, was that these TCP binding sites tended to be centrally located within accessible regions, within the center of accessible regions within this data set. And so if you take TCP binding sites from ChIP-seq in Arabidopsis and look at the accessibility of those, only about 20% of TCP8 binding sites are accessible. The rest are actually in nucleosomal DNA. So this is, again, data in Arabidopsis. But in maize, when we take our QTL data and ask, okay, if we can split these chromatin QTL that overlap TCP binding sites into variants with alternate alleles that either increase or decrease the affinity of the TCP to bind to this motif, what we find is that those that perturb the ability of TCP to bind always have a loss of accessibility. And so this can be also shown here, and that happens most frequently when the fourth and eighth bases of the motif are perturbed. Okay, so in all cases where TCP is predicted to lose affinity, right, by a variant that changes the specific sequence away from the consensus motif, right, where you would predict a loss or weakened binding affinity, in all cases, that happens, the accessible region loses accessibility. But you might be wondering, okay, well, why would you want TCP? Why, if TCP is controlling chromatin accessibility, but you see variation in accessibility across different cell states, how could you explain that? One of the things that we found is that the different TCP transcription factors show very dynamic patterns of expression within the family. And so to sort of summarize this part of the talk, I just want to say that we found that these QTL, the chromatin QTL, are enriched in TCP binding sites, and that these TCP, the binding sites from ChIP-seq data, right? So in vivo binding, most of them occupy, nucleosomal DNA. And then if we perturb TCP binding sites, right, if extant genetic variation affects these binding sites, they almost always exclusively lose accessibility. And that we can explain the variation in accessibility among different cell types in part by the cell type specific expression of TCP family members. All right. So what I've shown you so far is that natural variation, we can leverage this natural variation to understand, how genetic polymorphisms affect regulatory function within different cell contexts, and hopefully I've convinced you that the varying effects, particularly for chromatin accessibility, largely depend on cell context. Right. But one of the questions that we had coming out of this was, well, what about somatic variation? And so there's two members of my lab interested in answering this question. Fabio's a postdoc, and Andre, who's a research scientist. They wanted to know, can we estimate, the in vivo effects of a genetic perturbation on transcription, right? So somatic genetic perturbations. And so to do that, we found this system from it's been studied intensely by Virginia Walbot's lab. Brad Nelms is working on this now, and Damon Leash has worked on this historically, which is the MuDR system. All right, so this is an autonomous transcription or autonomous transposase that can recognize mu elements within the genome and mobilize them in maze. And so one of the nice things about MuDR is that it has a preference for either genic regions or accessible chromatin. So we reasoned that we could use this system to create or to use somatic mutagenesis to understand regulatory function in different cells. So the principle here is, can we compare cells with and without, right? The same, what we would consider the same cell type, with and without mu insertions in various regions of the genome. Can we measure the effect of a mu perturbation on expression using single cell genomics? So to do that, we generated a multi-ohm library from this material. So we're measuring the expression as well as the accessibility within the same cell. So these are paired measurements. And what we find, just sort of, anecdotally looking at this data, is that, we can separate out different cell types or different cell states pretty reasonably using just the RNA modality or just the accessibility modality. Although both of those technologies, they have trouble resolving some structure. So this large group of this population of cells from the transcriptome data, this population of cells from the ATAC data. But if we do a joint clustering, then we can resolve at a finer scale the different cell contexts that exist within the seedling in this case. So this just shows that we can identify different cell populations using both of these different modalities, but you get a little bit more information and more resolved cell context if you can do joint clustering and leverage both of this type of information at the same time. So the first thing we asked, because this technology allows us to ask some new questions, right? Because we have expression and chromatin profiling from the same cell. The first thing you can do is, can we identify, the regulatory regions that are associated with gene expression, right? It seems like a simple question, but you'd be surprised that, oftentimes, or at least in past studies, we have examples of long-range regulatory interactions between enhancers and promoters. Sometimes they can skip genes. So it's not that trivial of a task to try to assign a gene to each regulatory region. So the first thing that we did was to collect all the accessible regions within 100 kilobases of all the expressed genes within the maize genome, and basically just asks, which accessible regions are associated statistically with the expression of any given gene, right? So essentially, we're trying to fit as a linear model where the presence of accessibility or absence of accessibility, is it associated with expression, right? So we're looking for correlations. So by and large, most of these pairs, we find about 60% of all gene ACR pairs are significantly associated with each other, so more than not. And if we look at the distribution of those effects, almost all 80% of ACRs have a positive effect on expression, which we know, right? But there's also this 19% or 20-ish percent that are negatively associated with gene expression. So that's this blue example here, where the gain of accessibility is correlated with the loss of gene expression. So that would suggest that these are acting potentially as silencing elements. You can also look at the distribution of the number of ACRs associated with each gene, all right, split by whether or not they're activating or silencing. So if we look at just the activating ACRs, we see that on average, each gene has roughly 9 or 10, 8 or 9 ACRs associated with it. Okay, so that indicates that gene expression regulation is quite complex. Okay, it's not just involving one single region when those regions tend to have an activating property, but we can also look at the distribution of these silencing ACRs, and actually more often than not, it's only a single silencing ACR, which suggests that maybe these have stronger effects that may override, these positively associated ACRs when they are present. And so this is starting to allow us to pick apart the regulatory architecture on a gene by gene basis. So the next thing we ask is, okay, what about are there distance effects? If you're closer to a gene, do you have a stronger effect on expression? And the answer is, in general, yes. So within about 5 kilobases is where we see the strongest effects or the strongest correlation between accessibility and gene expression, right? And after that, it all sort of levels out. But in general, we still see uniformly strong effects, sort of regardless of how far away the ACRs are, and you can explain that through chromatin interactions, such as chromatin loops or contacts being made from distal locations. But in general, these long range ACRs tend to have weaker effects than those that are somewhere within 5 kilobases of the gene that they're associated with. And then because I had to look into this because of all the literature on super enhancers in mammals, and so we asked, okay, well, is there an effect of ACR size? So do bigger or larger regulatory regions are they associated with a greater effect on gene expression? And the answer is not really. There is a small, really weak correlation. It's about 0.2, the Pearson correlation coefficient, but by and large, they're pretty uniform. So no real clear super enhancers, at least within maize in the tissues that we've looked at. But one of the interesting things that this allows you to do is contrast the specificity of accessibility with the specificity of gene expression for those associated ACR and gene links. And this is where we do see a somewhat stronger correlation, particularly within this initial part of the range. So things that are maybe not very specific, right? The accessible region that's not very specific to any given cell type tend to not have very specific gene expression. And you see as the specificity of accessibility increases, so does the expression until you get to about 0.8 specificity. This is on the tau metric. And then the expression or the specificity of the accessible region just plummets. And so here's an example of one of those, right? So we have very specific gene expression, right? So this is a gene where we only really get transcription within a single cell context, but this region of the genome is accessible in every single cell type. And so these are, to me, are very interesting, and we're trying to follow up on this experimentally to figure out how regulation is happening despite the fact that you have pretty uniform accessibility. So what are the different transcription factors that are present? Are there other chromatin signatures that we can use to sort of elucidate why we only get transcription within this single cell context? All right, so I haven't really told you about the somatic mutagenesis yet, and so let's let's dig into that. So the first thing that you need to do if you're gonna try to leverage somatic mutations to understand mechanisms of transcription is identify where those mu insertions are. So we reasoned that we could use the clipped reads portion of the attack data to pinpoint reads or sequences that didn't have uh, anything corresponding within the reference genome. So here's an example of the mu-DR. We also generated the same data type in B73. Okay, so here's the mu-DR genetic background. We can find that this mu sequence here, was clipped in order for these reads to align. So that was just an example of a homozygous mutation where all the cells contain, or a germline mutation where all the cells contain this mu insertion, and we don't see anything in B73. We can also see observe some heterozygous mu insertions. So again, these are germline inherited, but the more interesting ones to us are these de novo insertions, where only a single cell or a handful of cells contain or exhibit a unique mu insertion, right? And so again, here's another example in B73. We don't see the same mu insertion. So we can look at the distribution once we have identified all these instances throughout the genome using all the cells within this data set. What is the distribution of mu insertions across cells? So those that are, most of them are very low frequency, as you would assume, right? A de novo transposon insertion would exhibit. But we do observe, little peaks right at about 0.5 as well as at 1 that correspond to parental heterozygote and a parental homozygote. And of course, you can get a larger range of the distribution of insertions across cells because of things like mitosis, where you get a cell division. If you have a mu insertion that happens early on in development, that mu insertion can propagate throughout the plant through mitosis. But by and large, scanning through this data, we find about 4,000 distinct mu insertions that we required to be, to meet our criteria for the following slides, identified in at least 2 distinct nuclei. And this number is way less than what has been observed empirically using something like MuSeq, in particular from Brad Nelson's lab at UGA. But we feel pretty reasonable that this is an underestimate, but we really wanted to be conservative for the following analyses. So just like the comparison we made earlier, one of the things that you can do is to ask, does a mu insertion within a regulatory region have an effect on expression, right? So now we're not just looking at a correlation, we're trying to ask, does a mu insertion affect expression? Again, this is through statistical approaches, but starting with about 12,000 insertion gene pairs, so again, restricting, measuring effects within 100 kilobases of an expressed gene, we find about 1,000 of these pairs are significant for a change in expression. So here's just one example. And we're showing you the volcano plot of the fold change between the cell that has the insertion versus the cell that contains the reference allele, right, with no insertion. So by and large, most of the mu integrations have a decrease in expression. Of course, we do find some that increase the expression, but this proportion is about the same as the distribution of ACRs and their effect directions. And what's also interesting is that we observe absolutely no effect on the effect size due to distance. So it doesn't really matter where mu inserts. If it's inserting within a regulatory region, you get pretty uniform effect sizes regardless of how far away the insertion was to the TSS. All right. So I just want to wrap up with just sort of a short discussion of how we think about cell type versus cell state, and thinking and coming up with this talk. I went to the literature and I found this definition. It's an informal definition from classical mechanics, which is a field of physics, for the definition of state. Like, what is state? And this definition states that a set of variables describing a system which does not include anything about its history. And this kind of struck a chord with me because, when we discuss or we talk about cell types and cell states, there is a very inherent, I guess, obligation to include the history of that cell, right? Like, what was the lineage of that cell? And that plays a remarkable role in what a cell type is, right? Where it came from. And so one of the things that I thought about is like, maybe we shouldn't really be using the word cell state because this definition would suggest that there are infinite number of cell states. And just bear with me, I'll try to wrap up here real quick. But the environment, for example, creates many different types of states, even transcriptionally within the same cell type, right? So, the plants in our growth room may be more stressed than the one in the growth chamber next door, everything else being equal. That's going to create a small change in the cell state, in the amount of transcription, the levels of transcription, even differences in shading, right? So, two different plants, one maybe slightly more shaded. That's going to affect chloroplast gene regulation and the output of photosynthesis, which has downstream effects on the rest of the physiology of any given cell. That's going to affect the cell state, where the plants are, what the elevation is, what they're experiencing mechanically, right? How much light is available, the nutrient or water availability. So the same genotype in two different labs, two different locations will be totally different, right, in terms of their cell states, and it's infinite. And another one that I think is probably pretty obvious that we hope to start addressing soon is even spatially, right? So you have, two different cells of the same cell type that are in two different spatial positions. They sense the environment differently. They're experiencing different cell-to-cell communications. That creates small variation or large variation in transcription, which manifests as a different cell state. And then, of course, somatic and germline mutations, i.e., the genotype, has a very strong effect on what is happening within the cell and what the cell looks like. But I would say that at least with multicellular eukaryotes, that using cell types is useful because we can define them. I can communicate to you if I tell you that the mesophyll cells, when we subjected them to salt stress, just completely shriveled, you would know what type of cell I was talking about. So it's communicatable, and they're generally more finite, right? But they do, of course, entertain the idea that, of course, they're quite dynamic. But at least, when we're discussing what's happening at the cellular level, having some sort of label helps with communication, and I think to me that's a useful characteristic. Um, Although I also state that being able to contrast cell states is also still useful, right? Because if I was to compare again different treatments, all right, those are two different cell states for the same cell type. So being able to communicate, right, what's happening at a cellular or molecular level with a label that's understandable to a broader audience, to me, is still a useful consideration. All right, so I know I complained about, don't use cell state. And yeah, I'm a hypocrite because, a recent paper included the word cell state, although I would say that we're trying to be a little bit more careful about our nomenclature moving forward. And then I just want to end with sort of what I see the outlook for with like single cell epigenomics. I think it's becoming pretty obvious that chromatin accessibility is easily profiled with all the commercial solutions, although it's still not perfect. We're limited by the number of chromosomes within the nucleus. There's still some uncertainty with sampling. Is the absence of a TN5 integration really the absence of accessibility? TN5 has its own issues. It has a strong preference for certain sequences. And then what's missing? Like some of the things that we would really love to start including is on a single cell level, transcription factor binding. Chromatin accessibility is nice, but we really want to know who's there versus who can be there, right? Which transcription factors are actually present and affecting transcription. What are the quantitative effects of CREs on transcription? We're starting to get at that with the mu work, but we need something more high throughput to really get at all the different perturbations. Of course, histones and chromatin information, as well as chromatin looping. There's some new techniques available now to do single cell Hi-C and things like that to get at how chromatin organization affects transcription. And of course, cell lineages. So this is something empirical cell lineage, not just pseudotime. We need proven proven, mitotic maps of what's happening during development in order to better understand transcription regulation from our perspective. And then, of course, perturbations, environmental, chemical, genetic, all the different perturbations and how these different contexts affect transcription. We need maps of that. And then lastly, real-time measurements, like what is happening in vivo in the cell in real time without killing the cell. I still think we're quite a ways away from that, but this is really the dream here. If we can get to real-time measurements, we can really start to make progress. And of course, I have to thank a lot of people from my lab, in particular, Fabio and Andre, as well as Lu Guang. Of course, Bob, my advisor during my postdoc, and many people from his lab and our collaborators and funding while we have it. And with that, I think I ran a little bit over, but now probably time for questions, right?
Yeah, thanks, Alexandra. We yeah are a bit over, excuse me, but I do want to get to a little bit of discussion. So there is a set of questions that we've been asking all of the speakers in this webinar series. And I think both of you have actually addressed all three of them, but I just sort of wanted to stipulate them again. Why, or I guess, is it important to define cell types or to, belabor this definition?
I'll let Elliot go
Um,
first.
Until we have a better one, I think we have to. But we know it's not encompassing what we really want to know. I mean, as Alex very correctly pointed out, that the RNA expression patterns and protein expression patterns are mathematically a manifold, and so they grade from one cell, a stem cell, to a differentiated cell entirely, and there are an infinite number of possible variations, not even including the abundant amounts of noise. And so it's the cells themselves are so far smarter than we are.
Yeah, I can't really argue with that. I mean, the thing that's, I think it's still useful as I tried to highlight, right? At least in terms of communicating, right? I mean, we still have to talk to each other and, express what we're finding in our results, and it has to be understandable to a broad of an audience as possible, if you want people to read it at least and understand it. So I think, we still need that definition, but I think it's getting fuzzier, and you know, this has been addressed technically, I think, through several efforts, including Plant Cell Atlas, to come up with a uniform set of guidelines and and ways to to have a best practice, I guess, so to speak, in how we communicate and what we use to communicate what is the cell type. But we're still not quite there. I think we're making strides, right? But even within my own lab, there seems to be different practices happening, and trying to rectify that is, I think, that's our first goal. But yeah, as long as we know what each other is talking about, then I think we're in the right direction. But as data sets get bigger and there's more and more adoption of these techniques and other ways to profile what's happening in distinct types of cells, it's going to become more important, that we really do have a set of restricted guidelines that people follow.
At a minimum, it would be useful for people to say when they talk about cell type what their assay was, whether it's RNA, protein, morphology, uh, stage in cell cycle, and so on.
mhm. Yeah, just have like a cell type as defined by X.
Yeah,
Um,
Yeah.
yeah.
that would at least let people know what you mean. Although, the entire concept's really not doing it for us. We need some new way of thinking about this, which we
Yeah.
haven't got.
So I guess that leads to the next question is like, how are you operationally defining cells and cell states, cell types, in your groups now?
I'll go first on this one. I think I kind of mentioned it a little bit. We're using marker genes, right? Like I think like most of us are who are working with multicellular species at least, because that's really all we have, right? We're really relying on the decades worth of preceding work of others who spent a lot of time doing RNA in situ and fish experiments to look at expression patterns of individual genes, and they've been really helpful. Part of the reason that I was drawn to Maze initially was because there was a lot of data from Sarah Hake's lab and Dave Jackson's lab generating lots of very useful marker genes that were super helpful for doing single cell experiments initially. But I think as we generate more data, moving forward, I think we got to start using other data, like the published data that's out there, instead of just using marker genes. Like, we need to start integrating and use models that not don't just rely on a handful of genes, which can be deceiving, as I learned through hands-on experience, but trying to integrate all the existing data that's out there. And that way, I think you can really come up with a uniform approach to annotate the cell types that exist within your data, or to try to identify the types that haven't been annotated yet, as we have seen from recent studies. That would be my take, at least.
Well, nothing to add. I mean, in terms of Calurpa, none of these concepts pertain. And so, and so I don't even know what to say. In our Arabidopsis single cell work, we do the same, more or less useless things that everyone else does, which is we recognize a very large multi-dimensional space that have clusters in it that you can show in these UMAP presentations, which are totally aquantitative, but they at least indicate that a principal component analysis can show you that there are more occupied and less occupied areas of that multi-dimensional space.
Yeah, and I guess like how can we do better? How can we like what does the future look like in terms of definitions or you
Well,
know
look, people have been talking about these sorts of things like, for example, the concept of an dedifferentiation, an undifferentiated cell. People have been talking about that for 120 years, and no two people have ever meant the same thing by it. So I suppose we've got at least 120 years to sort this out. It would be better if we tried to achieve some sort of clarity.
yeah.
Yeah.
Like Potter Stewart's definition of pornography, right? I can't define it, but I know what it is when I see it. And that's kind of the realm developmental biology has been in for since the 1890s. I don't know why, genomics could, shouldn't take that as a proper example, but it's not going to destroy us. We can't figure this out soon.
Mhm.
Yeah, I'm going to take a really practical answer to this, mostly because I want my students to follow these principles, and that's to use again all the data that already exists. We don't need, if we can generate spatial transcriptomics data, great, right? Because at least there, I will say practically, the bottleneck with single cell experiments is annotating. It's figuring out what is the identity of the cell, even loosely, right? Because it's really hard to make inferences biologically if we're looking at, a bunch of ATs, Cs, and Gs as the names of our cells. What we really need is something that we can attach more meaning to. And so, I think we still need more data, ground truth data, and I think spatial transcriptomics will play a big role there, at least in helping to anchor a lot of the single cell data sets that already exist. Even just for that, to be able to define, okay, well, this single cell data point has a very close match to this spatial position from a spatial transcriptomic map. I know this is very basic, again, practical solutions to the question. I'm not going to, get into the philosophical aspects of it because I'm not qualified, I don't think, to get into it. Not a developmental biologist by my own admission, but I still see the value of having labels and being able to make contrast, because it does simplify some of the ways that we can ask questions. But that's not to ignore that, cellular diversity really is, I agree, on a gradient rather than, something that's really well defined, because they're not. Because it's
Yeah.
biology, there's no rules really.
Awesome. Well, thank you. I think we've blown way past the time that we have, which I mean I think is great. I think there's been a lot of really interesting science and some discussion. I wanted to yeah, close it up by thanking both of our speakers today, also Natanella and Trevor for organizing this entire webinar series. In a couple of weeks, June 10th, we're going to have another set of speakers, Marina Olivia and Alex Cantopastor, who will, yeah, I'm sure present some really exciting stuff. Yeah, there are, I think, some open questions on the Padlet. I am going to start entering some of my own because I have a lot of follow-up questions for both of you. But yeah, thanks again, and thanks all of you for attending. Tanella or Trevor, do you want to say anything before we close out?
Just thank you as well for moderating this
Yeah.
interesting.
Thanks, Ben.
Yep.
Thanks, Ben. Thanks, Alex and
Right.
Elliot.
Yep.